Okay, you see this? This is the slide, right?
No, Prof, I can't see.
Not yet. Okay, hang on. I tried to share, but it didn't come out. Share. Okay, got it. Yeah. Okay.
Yes, Prof, yes.
Okay. And I need to jump here and there because of the situation here. Okay, I think about this these slides, right? Direct examination, enzyme link immunosorbent assay. This actually a test that we use, to measure antibody. It is highly sensitive and then it is specific. So enzyme link immunosorbent assay test, acronym as ELISA. So measure the concentration, specific antibodies to detect the presence of various what we call these antibodies, as well as what we call this protein, various type of protein. It's very sensitive. In fact, they use this technique not only to detect antibody or protein, also hormone. Hormone are proteins anyway, so they use it. So it is highly sensitive, and then we use ELISA as a method to measure antibody titer, how much antibody that you have in the serums, of the sample that from the host. Does not matter from human or animals, so you can use the same test. What is that? Two participants waiting in the lobby? I don't understand this.
Prof, just approve them, Prof.
Oh, just approve. Oh, that's what you mean, huh?
Yes.
Waiting in the lobby, but I don't know where to approve. Allow attendees to.
Usually there's an option at the side.
Where to find it? Where to find that things? Why I have to approve that? I thought it's already it's already
Prof,
it's already
sorry?
it's already automatically. I mean, it's automatically joined. Where are those? Okay, participant lobby or two of them. Okay. Okay, that's what I mean. Okay, in the lobby now already here upstairs, no more in the lobby. Okay, this is actually about the ELISA test, nah. So basically the ELISA the step of doing ELISA, it is not that difficult, but it is of course it takes some time lah to prepare. So the principle is still the same as drafted here, general step here. So if you have, let's say, this actually the the 96 well, I make it big. So first of all, you add the antigen. So add antigen. Oh yeah, sorry. Add antigen here. So antigen, whatever here, you add antigen here. You code. You make sure the antigen actually attached to the plate. Add antigen. Then there are many antigen because you add antigen in the form of solution. Sometimes you have a extra antigen on top of it, then there is a washing step to remove whatever excess there. So excess means that extra and protein that you would like to test. Assume that, let's say, you want to test against for the presence of herpes virus, equine herpes virus, So you use equine herpes virus, and then you add into the plate equine virus protein. Yeah. Then after that, you add serum, the test serum. So the test serum should contain antibody. Okay, antibody here. So if they are specific, they will bind to what we call it, to each of the antigen here. So they'll bind. They bind to the antigen. So if let's say they are not specific, let's say the serum does not contain antibody, meaning that this what we call this binding won't happen, meaning that no antibody will bind to the antigen. So this is actually the equine antibody. Then after that, whatever, let's say, excess antibody, you throw away again, because if you leave it there, it can cause a cross-activity. Then after that, you add another antibody anti of this antibody, anti the first antibody. So the first antibody is a test, which is inside the serum. This one is actually anti-equine antibody. It's already linked, this antibody already linked with the enzyme here. So we say this antibody linked with the certain what we call this enzyme here. We carry enzyme. The antibody here can be, you can do something in the lab so that this antibody with the enzyme. This enzyme is actually more is a normally peroxidase based, Then after that, you add, let's say, chromogen. Chromogen is actually color. This what we call this chromogen, it is colorless. But if there is, let's say, if let's say this chromogen react with the presence of the what we call this enzyme here, so this will show a color. Okay, so become color lah. Okay, color. So if let's say there is this antibody, they don't bind to this antibody, so this chromogen, or we will call it substrate, cannot what we call this react with what we call this enzyme. So there is no color. What does it mean? Now, if let's say you have a lot of antibody bind to this antigen, so the color, the enzyme link antibody bind to the first antibody also become many and many. So the higher the antibody here you have, so the amount of, let's say, this antibody to bind to this one also increase. When they bind the amount of antibody bind to this first antibody increase, the color intensity also increase, meaning that becomes stronger. If it is red, become red and more red. If it is green, becomes from light green becomes very dark green. So the intensity of this color can be measured by using spectrophotometer. Okay, there is a specialized machine that uses a spectrophotometer there. So in this case we call it ELISA reader. ELISA reader is actually spectrophotometer to measure the color penetrations, the light penetration in that particular color, the spectrum. So the what we call, the higher the antibody, what we call level there, the darker the coloration, then the higher the what we call this OD, optical density of the light that cannot go through that or absorbed by the what we call the solution. That is a general step. So you read this one. I hope you read this one, because if we ask you what is the principle of ELISA test, this is the one, very basic. This is actually basic, but ELISA you can modify it, for different way. But the principle is the same. This one can be direct ELISA, indirect ELISA, sandwich antibody ELISA, double sandwich, whatever, competitive ELISA. There's some variation in terms of the use of what we call this antibody antigen, but the principle is this. You have antigen, you have a test antibody bind to the known antigen, so meaning that this one is known layer, known antigen. So as I mentioned, assume that you want to test, for the presence of antibody against equine herpes virus. So you need to put the coat the plate here with what we call this equine herpes virus protein. The unknown, the test is serum here, the second layer. So the third layer is actually the one that going to give signal. Yes, the antibody is here. So that's how it works. Okay. Okay. There are several other methods here. Just to tell you. Complement fixation test, hemagglutination inhibition test. Okay, hemagglutination inhibition test, I don't want to do it. I don't want to explain here. You can you will learn here with, I think, with Dr. Yasmin during the practical session. Same thing with the immunofluorescence. Same thing with the neutralization test. So neutralization test is simple. Antibody and virus, they bind, they neutralize each other, then that's it. The virus cannot attach to any cells anymore. So that is actually the principle of neutralization test. The virus has been neutralized. What does it mean? So other technique, actually they use radio immunoassay. This one is they use iodine, what we call this radioactive isotope iodine and so on. So EIA, enzyme link immunoabsorbent assay, is actually another word for ELISA. Okay, enzyme link. So sometimes we call it EIA, sometimes they call it ELISA. And this one is a Western blot. Western blot is actually a different method. Normally, for detections of what we call this antigen, the principle is the same, but instead of using the plastic, we use a paper, blot paper. So blot paper, we put antigen on top of this. Okay, put the antigen or the protein on top of this. Then we add the test serum here. Test serum here. Then we add, let's say, another layer there, just like, let's say, ELISA, which is what we call this enzyme. Antibody already linked with the enzyme, and add what we call the substrate to make it color whenever this positive. So positive means, yes, there is a reaction. the antibody is there against whatever disease agent that you tested. So this is actually the principle here. You go back and read carefully. You don't understand, you ask me. Sorry for this one. I didn't save it. Shouldn't be here. It's a repeat for this one. I need to save this one. So this is a Western blot. Virus isolation, we're talking about to get the virus from the cell using primary cell, monkey kidney, but you need to prepare this, you need to obtain tissue from monkeys, from the animal, which is not really what we call this, to me, the best method, because not every time you will have to do the test, virus isolation, you have to kill the animal to prepare this. This one you need to prepare. But this one, continuous cell, readily available. You can get it from what we call this commercial supplier. So there are various types of continuous cell. And then the origins of the cell can be from many species. HeLa, human, Viro, monkey, SCP2 from human, MK for monkey kidney, monkey cell. MDCK is actually canine cell, C for canine, Madinda B canine, kidney cell. So it is not the issue if you use a continuous cell line. Cell culture, I mentioned yesterday, I think, about the cytopathic effects. You can see the ballooning of one of the what we call this cytopathic effects, actually the pathology that occur in cells following virus infection. What you're going to see, example, ballooning, the cell become rounded, syncytial formations, because the cell actually fuse together, fuse 2, 3, 4, 5 cells fuse together, become multi-nucleated cells. So we call it syncytial cell. Or the formations of inclusion body, or formations of vacuole inside the cytoplasm. The cell not growing anymore monolayer, become multi-layer. Okay, and it can be very specific, it can be non-specific. So hema-absorption, blood cell, the cell that acquired ability to stick to mammalian cell. Normally, cell culture, they cannot bind to red blood cells. But following virus infection, virus infect the cell, virus will produce their protein and put it on the plasma membrane of the cell, and following that, the cell, the infected cell, can bind red blood cell, erythrocyte. Okay, so these are the things that it can be done. Various things that it can be done. This is a part of the what we call this CPE developments of the CPE. Next one is you have seen this one previously about the cytopathic effect. I show you already. This is what we call this ballooning of cells, formations of plaque here, one plaque here, this one another plaque here, plaque formations. You can see the cell here are ballooning, become rounded and inclined to detach from the plastic membrane, the floor, or where the cell used to attach. This actually is the syncytiae, okay, big looking cells, but actually is following the what we call these fusions of several cells together and becomes one. Why they fuse together? Because when virus actually inside, so they produce, let's say, protein on the membrane, then this actually protein belongs to the virus. And remember, virus actually envelope to enter the cells, they actually fuse with the cell membrane or plasma membrane there. So same thing happen between 2 or 3 or 4 5 different cells, because they already have the virus protein. They can fuse together because of that. Okay. Problem. What? This another one here. I think the same thing here. I already mentioned. Problem with cell culture. Long period acquire for the result. Sensitivity is very poor. But you need to do this for virus isolation. It's not the issue. But if you rely on the CPE only, then it's going to be a problem. You have to do more than that. And then the problem with the cell culture itself, is actually very easy to get contaminated with bacteria. Because bacteria is also cell, they can grow very fast, faster than cells, because the medium cell growth medium for the cell actually very, very good for the bacteria to multiply. And then specimen may cause a toxicity to cell culture, then cell will die. Instead of, let's say, virus cause the death in the bacteria, the toxic substance also can cause death to the cells. So what are the toxic substance? The brain tissue, if you, let's say, homogenize and so on, and then put it in cell culture, this brain tissue actually toxic to the cells. And then another issue is not all viruses can be grown in cell culture. Not all. Many viruses, in fact, cannot be grown in cell culture, especially those viruses that normally cause diarrhea. Parvovirus, okay, papilloma virus cannot grow in the cell culture. So rapid culture technique. This is actually what we're doing is actually like this. Why we do rapid culture technique? Because we don't want to wait for the virus fully infect, let's say, the cell, grow inside the cell and release the virus. Because now, if you're dealing with, let's say, slow growth virus like CMV in this case, then it may take, let's say, 1 week or 10 days or 2 weeks, for you to see the result. So one of the ways is actually you infect, let's say, the sample that may contain the virus into the cell. So within few days, the virus already inside the cell. They grow it slowly, but you couldn't see CPE yet. Then you can do the test on these cells by doing staining, by doing whatever. So one of the method that normally we do test this one to check for the presence of viral protein inside the cell. We do the immunofluorescence. So you learn this how we do fluorescence later on. So immunofluorescence we do to check for the presence of the antigen, viral antigen in the cells. So cell from where? Cell could be from cell culture or cell from the tissue itself. Can be either one. So in this case, if you want to get the, let's say virus sample in the solution, you can test for the presence of virus by putting them into the cell culture first. That's what I mentioned first, and then no need to wait for 10 days. Within few days, the virus already growing. Despite you cannot see any CPE there, but they are there. So you do this kind of technique, you can see the detections. The virus can be detected in that way. So virus isolation there. Not to mention this one. As virus isolation in hand aches, normally if it is pox virus, we use CAM. Newcastle disease virus, we go for chorioallantoic cavity. If it is influenza virus, we use chorioallantoic cavity. Okay, this electron microscopy. Okay, one of the issue if you want to do EM, electron microscopy, you need to have this concentration, virus concentration. So we need to have 10 to the 6. You're talking about a million virus particle per milliliter. A million, 10 to the 6 virus particle. From the sample, direct, let's say, you cannot get this normally. It's very simple. Mungkin if you can get 1,000 virus particle, maybe 100, it's too few. That's why you cannot do EM. So most of the time, you need to grow the virus for your cell culture, get 10 to the 6 virus particle per mil, then we do the electron microscopy. Except for certain viruses like rotavirus, this one. Rotavirus, parvovirus, this one, so corona. You don't have to do what we call this cell culture, because some of these viruses cannot be, you cannot grow them in cell culture. You can use certain technique to make them concentrated, get let's say the sample as much as possible, do a technique to concentrate the virus first by using the centrifugation and so on. Then from there, you can do the EM, electron microscopy. Also from the vesicle fluid here that contain large amount of virus, or from skin scraping virus inside the cell, or virus, pox virus, and so on. And also from the blood, swine fever virus. Now, swine fever virus in the blood, you can see that lots of virus particles. But still, subject to 10 to the 6 virus particle per mil. So, because this sample contains a large amount of virus, then it shouldn't be a problem. So this I already tell you about this. This about the problem with EM, expensive equipment, expensive maintenance. You need to have good experts in this area, and sensitivity of low. Low mean that you can only detect, let's say, virus when you have 10 to the 6 of virus particle. If you do PCR, you only need 1 virus particle to do that. You don't need, let's say, 10 to the 6. Okay. This actually, when you do serology, okay, this is what you need to know. How you check for the antibody, okay? And you get the serum sample, you do the test, ELISA, or serum neutralization test, So how do you know this is a primary infection or reinfection? How do we know? You get the sample from the animal, or someone send you the serum sample to you to the lab. Test it. Please test. I suspect my farm is infected by this virus or whatever. Then you test. So how are you going to tell them and see whether it is a recent infection, primary infection, or reinfection, meaning that animal already long time already get the virus, and maybe they already recover from the infection, the disease, then reinfect again. So this is what we call reinfection. So normally, when you do test, you must have a standard for you to benchmark, for you to compare. You must make sure that one, you test, let's say, ELISA on the serum sample from a healthy animal or non-infected animal, okay, and compare with the serum sample from animals suspected infected by the virus. So you have to compare. If you don't make this comparison, then you don't know what is going on. Okay. So normally, if you have, let's say, 4 times increase of the IgG antibody titer, okay, uh, compared with the normal individual, that's it. This actually animal recent really infected. It's a primary infection. Okay, it's a primary infection. And then if you see large amounts of IgM, meaning that the animal is actually recently infected, maybe first time infected. Okay, IgM. Okay. Then you can see the seroconversion. Seroconversion means serum, conversions convert. Seroconversions means that animal produce antibody against that particular virus. So I can say that the animal is seroconverted against all virus, meaning that animal already produce antibody against all virus. That's what seroconversion means. Sometimes it can have to, antibody production, but seroconversion meaning that animal have that. Not to convert anything. Don't make mistake by that. Seroconversion meaning that the animal produce antibody against that particular virus antigen. Um, okay, one thing. If you, let's say, do the ELISA and you only check for the you from the serum sample, that system, you check, you get the antibody titer from the animal only. You see, oh, you can see that high antibody titer, IgG level here, high. This actually, that way, it is not that kind of approach, you're not comparing with anything. It is not reliable for you to make any conclusion. Okay? So you have to do benchmark here. So single antibody titer from the single sample, no comparison with, let's say, normal animal, it is not useful. You cannot say that. So, for example, here you see 4 times, fourfold. Fourfold of what? Fourfold of the normal animal. So, what about reinfection? Reinfection normally related with the rapid increase of IgG. Okay? Or total antibody acute and normal. Individual meaning acute meaning infected, normal individual normal. And absence or slight increase in IgM, slight presence of IgM. So let me show you the diagram. What does it mean? Okay. See here. Okay. This graph is very, very important when you go to field. So first time infection is like this. First time infection. So animal infected here. Okay. Then after, let's say, few days, normally 7, 8 days, 9 days, or 10, 14, you can see the IgM first. This is the IgM. Okay. Normally 14 days later, day 14, then you can start to see IgG here. So here, this is actually just what we call this visualization. You may not get it. In actual case, it depends on how good you are doing the test. It could be lower than this, okay? But the principle is still the principle here. You see IgM. IgM here, okay, normally very high among the IgM within first week, between the first week and second week, IgM here. Then after that, it reduces to the base. So reinfection, you still have IgM, but lower, very much lower than this. Still detectable, but very much lower from the first time. But infection here, infection here for IgG, first week, nothing there. Normally undetectable, very low. After second week, you can see the elevations of antibody here. Okay. You see they reach the peak at normally between 2 to 3 weeks. Then after that, going down. Why is going down? Simple. Going down because naturally when going up, immune system already remove some of the what we call these antigen viruses and so on here. Then due to homeostasis of our body, they don't need to produce extra, let's say, antibody against the virus, and then maybe you already start to recover here after 3 weeks, 4 weeks, and so on. So the antibody will go down. If, let's say, there is no reinfection, there's nothing there, there's no second exposure, this one will go down like that. Okay, will go down like that. Okay. So that's why this actually the basic principle of this one, of this. What is the basis of this? So for vaccination is the same. You give, let's say, vaccination here, looks like this one. But if you don't give another booster here, another injection here, you wouldn't get the higher level of IgG response. So that's why when you go to the hospital clinics, now you got for hepatitis B vaccination, hey, after 1 month, 2 months, come back and give you another booster, something like that. But you disappear, you don't get any booster, vaccination, you wouldn't get this high antibody, and you are still highly exposed to those viruses. So you go first time injection, you must go for the second time injections, because otherwise whatever you paid earlier is useless, because after a few months it goes back to the basic just like this. So in order to get this one, this is what we call booster dose. Booster dose, booster actually, BOOSTER, for the vaccination. So first time, second time. So second time you can see high antibody, and the antibody after that going down slowly, just like here, but it takes longer to go down. Okay, this is a complement fixation test. I'm not going to, you learn this one from the later on. I'm not going to talk here, not much time left here. Okay, you can read this one from the slide. Detections of IgM or rising titer of IgG in the serum, it is indications of the active disease, recent disease. Recent disease, okay. And then the problem with serology, longer period of time required for diagnosis for, okay, hang on. Okay, there is a terminology here. What is this? PET sera. Remember this. What is PET sera? What is PET serum sample? So normally, okay, you can read this one later, but I'm not going to. Normally, it's still related with this one, diagram. PET serum sample. So you don't know when is actually infection 013, but assume that, okay, assumption, you see the animal is quite sick. So you take serum sample here, first serum sample, assumption. You don't know on what day you already infected, because this one is already passed, and then you get this one actually from the farmer. So you measure here, wow, antibody here. So I got the IgG antibody. Then 2 weeks later, okay, 2 weeks later or 3 weeks later, you take another serum sample, see, let's say here, inject here. So you take serum sample here. So what happened? Yeah. So antibody increase. Okay. Normally 1 week or 2 week apart lah. So something like this one. So normally, in principle, if let's say the antibody here increase, so because of the PET cell sample, it's 1, 2, they call it PET. So it means that it's actually infection. Animal is actually infected because they have the active virus replication. Okay, and if let's say the antibody titer is actually the first one is here, but the second one is here, same level, meaning the antibody is actually high here. So the graph is more towards, look like, see. So this is actually normally related with persistent virus infection. Okay, but if you see this one, the first one is high, the second one is actually already going down. So the graph will be look like this. So this actually, the animal could be on the path of recovery. Okay, the 3 different scenario here. It is logical, it's logic only. So if the first, let's say, sample give low antibody, second higher than the first one, this is actually recent infection. But if it is actually high, same what we call this antibody level, first and second, meaning that it's a persistent infection or recent infection. If let's say the first is higher than the second, then meaning that the antibody level is going down, meaning that maybe the animal is actually on the way towards a recovery. So it is logic there. Okay. Okay. So problem with serology, you can read that problem. Maybe you can get the false positive results. Why false? I mean that it's supposed to be negative, but your test show that it's positive, but as a matter of fact, it's negative. Or maybe another way around is actually, is this supposed to be what we call this negative? It's supposed to be positive, but it show you negative because of the test. Something wrong you do during the what we call this sample processing, maybe the way you conduct the test in the lab, maybe the machine error, so you may get that. So how to address this issue? Normally we we do in duplicates or triplicates. Every time we do tests, we don't use only one sample, we use few samples, we split into 2 or 3. We would like to see whether is it sample error or the actual error, the actual is actually actual issues. Okay, so you see this one lah from lab. Just to remind you about the fluorescence. What else? Nucleic acid probes. This one you learn later about in the lab how you're going to do it. Okay, I think that's all. The basics about the normally the test that we do. Okay, the PCR is something that you need to learn actually. It this one the best I want you to make reading how PCR actually normally work, because if I want to make you visualize this one right now, maybe you cannot imagine that. Better go to the internet, check the YouTube, the principles of polymerase chain reaction for virus detection, the principles of PCR for virus detection. So it's very simple. There are lots of what we call these resources in the internet regarding the PCR. Okay, right. That's for the to recap that. That's actually the briefly about the what I'm supposed to accomplish in the previous lecture. So now we go for this one, the actual stuff, the actual things now about herpes virus. Herpes virus. Okay, what is herpes virus? Herpes. What does it mean, herpes? Herpes means creeping. See, okay? Yeah. Latin name of herpes, creep. Herpes virus is double-stranded DNA virus. Creep, herpes virus, okay? So what you need to understand the general characteristic of herpes virus, morphology, how the virus replicates, and so on. So herpes virus is very famous for the latency and reactivation. So if let's say you give example, if let's say that if there is what we call these questions on the latency, so please describe mostly related with the herpes virus. There, because of course there are other viruses also related with latency reactivation like HIV and so on. But normally it's easier to go for this herpes virus. And then human herpes virus, of course, no question there. But it is good for you to know because we all human. You go back, at least you can be a para virology. You know, if some, because there are so many viruses that we human are contracting from now and then, so you can explain to them what is actually virus is all about. Then for important viruses in common animal species, meaning that important herpes virus in common species, to see the disease nature, pathogenesis, vaccination, control, immunity. This is actually basically what we ask questions lah. The pathogenesis of the viral diseases, and then what are the methods that we use to control this disease from spread? How do you diagnose this? Okay, these are common questions that we ask. Okay, herpes virus, right? It is envelope virus, big virus. It's not super big, but it is among the big virus, It's 120 to 200 nanometer, varies, because there are many types of these viruses. From small one to the big one, okay? So the big one is it's a 50% the size bigger than the small one. So coronavirus is about 120 nanometer. So of course, COVID is not herpes virus. COVID is a coronavirus family. So herpes virus is actually envelope. Okay, remember herpes virus envelope. You see my face. You know already. Herpes virus is my PhD work. Okay, you remember me. You remember herpes virus. You remember it is envelope virus. So herpes virus, big virus like this. So you can see these are the envelope. So this is actually the capsid. So there is a space between the envelope and the capsid. So you have envelope, you have capsid, icosahedral in shape, and here is the space here, we call it tegumen layer. It contains viral protein for the initiations of virus replication following virus infection to the cell. This tegumen layer contains factors that can contribute to the initiations of the virus replications. The beginning of virus replication is a linear double-stranded DNA genome. Icosahedral is very easy. Yes, so that one is actually a trademark of herpes virus, icosahedral capsid. So linear double-stranded DNA genome. So we may ask you a question like this, okay? List, let's say, five common or basic characteristics or properties of the herpes virus. Maybe we don't say the herpes virus, we say that porcine herpes virus, equine herpes virus, what we call this bovine herpes virus. But how to answer that question? So when you say describe the properties of this or give list, let's say 5 or 6, 7 or 10, maybe whatever, basic properties of bovine herpes virus. But in general, it's actually herpes virus. You can use whatever info that we have here to answer that question in that way. So herpes virus, DNA virus, replicates in the nucleus. All DNA virus replicates in the nucleus, except pox virus replicates in cytoplasm. So replicates in nucleus and produce eosinophilic intranuclear inclusion bodies. Eosinophilic is actually more towards red color inclusion bodies. Under the microscope, we're going to see this. So tegumen contain viral enzyme, envelope contain glycoprotein which appear as a short spike under EM. So cannot even see here where are those spikes. This actually glycoprotein, viral protein. So again here we use the word glyco, sometimes protein, sometimes glycoprotein. Glycoprotein actually is a protein, but why we call it glyco? Because that protein has been glycosylated, meaning that during the virus replication, so the virus caused the protein to be glycosylated. Glycosylation meaning that additions of glucose or sugar onto the protein. So meaning that the protein has bounded to the sugar molecule. That's why we call it glycoprotein. So other than glycoprotein, you have phosphoprotein, phosphate element that bind to the protein. We call it phosphoprotein. But in this case, the one the antigen here on the surface of the virus on the envelope, all the time we call it glycoprotein. But if you read through books or journal, if they say that just protein, it's fair, it's enough, it's not a problem. Okay. Next. So morphology of herpes virus. See that? So even though under what we call this microscope, it looks like in the previous one, but there are variations. No, it can be looks like this, but in this case, no envelope, just capsid. Because no, this is actually purified. These are purified virus. When you purify virus, the virus will go through a certain process, and this process will cause envelope broken or and then eliminated from the what we call the solution. So you're going to see this rounded figure under the what we call EM. Then you may say that, oh, this is a new virus, not herpes virus. Okay, it's actually no, it is herpes virus, but because of the process during preparations of the pure virus, the virus may lost their envelope. So same thing here. This is actually the whole virus. This is the envelope, and this is actually the capsid here. Here this is the capsid. You can see that icosahedral shape, just like diamond-like shape. Okay. Oh, okay. No need to. I'm not going to go through that. Not there. Okay. How? Let's say, what is a virus herpes virus strategy to cause infection and to become latent? There are many viruses. Actually, human viruses, the way people study is very advanced. That's why I use a lot of human examples here. So many herpes virus cause lysis of the infected cell. So that's why I call it lytic infection or lytic infection. Lytic infection or lytic infection means that lysis of the infected cell. So that's why you see the cell being broken because the virus replicate there and to be released into the environment. And some, let's say, herpes virus produce tumor in human EBV, Epstein-Barr virus in human, Marek disease virus. This is the one that you have to remember. Don't forget this. Professor Rahman, actually, his PhD on Marek disease virus, and then he should have a more inclination to ask, let's say, to give you questions not related to Marek disease virus. It's a special virus anyway. The virus that cause tumor in avian. It is interesting virus. And all herpes virus, one is the characteristic is all produce latent infection. All of herpes virus. Does not matter alpha herpes, beta herpes, gamma herpes. All of them produce latent infection, meaning that latency, established latency, but where they remain latent maybe varies, okay? Maybe varies from one type of virus to another type of herpes virus. So basically, virus infections like this, virus infect, this is actually related, this schema, okay, related with the chicken pox in human. So virus, if let's say infect the skin, got through injury, replicate here, this area. As after some time they have enough, they will be transported into the peripheral nerve. Okay, peripheral nerve. Okay, transportation to peripheral. In the end, they end up in the trigeminal ganglia. Trigeminal, this is actually the nerve and ganglia. And then in ganglia, they become latent. They don't produce any virus particle. They only exist in the form of nucleic acid in the infected cells. For how long? For how many years? Until the immune response break down from the host, then the disease, same disease will appear again. Okay, you can read this one. Herpes virus divided into 3 subfamilies: alpha herpes, beta herpes, gamma herpes. So alpha herpes basically cause localized lesion of skin and mucosal surfaces of respiratory localized lesion, not localized what we call this replication. This is actually the cause fever and so on. Beta herpes virus, respiratory and generalized disease, and gamma systemic disease and tumor. Okay, so herpes virus eight groups in human, and many animal viruses, include, and also in many animal herpes viruses. You will find, let's say, in animal species, feline, canine, chickens. In fact, what we call this fish, you can see the herpes virus. It's everywhere. And genome, this is what we call the genome mapping. I already told you before, this 2 lectures ago. We come up with a map of the genome so that it's easy for us to, it's easy for us to visualize which gene that we're talking about, where is the gene, how, let's say, the gene related with other genes in the system. So that is why we use what we call this genome map, to sometimes do the virus classification too. So based on this mapping and so on, many herpes virus also, they got new grouping, new name and so on from time to time. Okay, okay. One thing about this, what we call this herpes virus carry in the tegumen, part of tegumen, one protein, here, which is, we call it VP16. No need to remember this, So what I'm trying to tell you, how they replicate here, when, let's say, the virus enter the host cells, so they immediately, what we call this, virus will produce, we do the transcription and translations of immediate early genes. So when immediate early genes activated, so they have immediate early proteins now. This immediate protein will cause the productions of early protein encoded by early genes. Okay, early genes, of course, produce early proteins. This one will promote early genes. Then the early genes will, let's say, promote the productions of this late protein. This actually this way, right? So actually, early gene will promote immediate early genes will promote the productions of early genes. Early genes will promote the production of late gene, but late gene will suppress early genes, and early gene will suppress IE gene, so that this one won't be continuously be produced at higher amounts. Suppress means that suppress production. They reduce the amount of production. General features of this virus. Epidemiology, they are not stable. Survive for only short period in the environment, in hours, And the transfer of this virus need close and intimate contact between animal for transmission. Not like, let's say, COVID-19, sneezing and so on, you have, let's say, particles, not like that. This one, you need to go for the close contact. So they have latency actually to ensure the survival of the virus and to make them persistent in the environment. How do they know that? I cannot answer that question. How come, virus can decide what they need to do? In order I want to be here for a long time, I generate capability to become latent. I cannot answer that question, how the virus can do it. Is it in their genetic code? I'm not sure about that. It is what the, let's say, the science, nature telling us about. Latency is a very important topic in herpes virus. It is actually a mechanism for virus to remain survive, for a long time. So acute infections mean that here, acute disease, acute infection is a situation where first time, let's say, the animal or host actually exposed to the virus and produce a disease. Okay, that's what we call it, acute disease. Then after that, the virus now, remember, transported into certain places, especially intrageminal ganglia, and remain latent there for a long time. Okay, some of these herpes virus also can remain latent in the lymphocytes, but common one here, ganglia. Upon, let's say, stress factor because of training, whatever, fever, surgery, whatever, so the immune systems of the host becomes compromised, weakened, So as a result of that, the latent viruses will be converted to the active virus replication, and following active virus replication, that's how you're going to get acute disease in these animals. So next, so this is actually what you see in human, this one is about the human herpes virus type 1. It's a herpes simplex virus. Okay. Example. Okay. This one is chicken pox, the one that you know. First time infection, we call it chicken pox, or we call it also varicella virus. But the same virus remain latent after some time. They reactivated, then to produce a disease, recrudescence, and that disease we call it shingles. Chickenpox and shingle, they caused by the same virus, but the difference is the first one is acute infection or primary infection. The second one is actually reactivation following latency. So they just to show you just example. No, okay. How, let's say, the virus establish latency? Because of the work of let gene that only occur present in herpes virus. So let gene stand for the latency associated transcripts. Okay, during latency, only this transcript you can find present in the cells. So that's why I call it. And then this actually latency associated transcript encoded by certain genes in the virus genome. So that's why I call let gene too. So I mentioned this one. Let what we call this transcript, mRNA here, are the IE gene, and then so that they can be detected during lytic infection, but are present at much higher level. Meaning during latency, let gene level is high, active infection low. Okay, this is actually example EHV1 actually latency in trigeminal ganglia, EHV4 also trigeminal, EHV2, EHV5 also trigeminal. But there is another place, other than trigeminal. EHV1, T lymphocytes. EHV4, lymph nodes of the respiratory tract is a place for them to remain latent. EHV2 and EHV5 actually both actually gamma herpes virus of equine. So they also can go latency in B lymphocyte. Okay, this one I'm not going to go through this lah. It's about tissue culture here. You can see that alpha herpes virus highly cytopathic culture, causing rapid destruction of susceptible cell. Okay, whereas this one gamma is actually is specific. Can infect, let's say, B cell or T cell lymphocyte, and then the gamma they have oncogenic, may cause cancer. And then the alpha herpes virus actually is the fastest grown virus compared to other viruses, beta herpes virus or gamma herpes virus. Okay, I'm not going to go through this. This actually human. You can see the in cattle we have a bovine herpes virus one. Okay, this is interesting. In almost every species, you have herpes virus. From mammalians, avians, ikan, the fish also have herpes virus. And the nomenclature of herpes virus, they always have herpes virus 1, 2, 3. And actually, most of the time, bovine herpes virus 1, equine herpes virus 1, even feline herpes virus 1, all actually herpes virus 1 actually related with, first, they are related is, a common indicator is respiratory disease. So they commonly cause respiratory problems in the susceptible host. Okay, you need to read this one on. This is actually a special assignment for you. Hopefully, it will come out in your exam. Infectious bovine rhinotracheitis, IBRV. Okay? If, because this actually the scientific the generic names of the virus, but the scientific name accepted by ICTVM is actually BHV1, bovine herpes virus 1. It causes 3 different diseases here: respiratory, reproductions, okay, and also nervous system here. Okay, so 3 different systems. So they cause problem in, let's say, respiratory, reproductive, and also the what we call this nervous problem to a certain extent. You read this one. So another name for this, you have infectious bovine rhinotracheitis. This is actually the common name being coined by a clinician, 20 years ago. Instead of BHP1, they mentions about IBR and IBR. It is actually the same virus anyway. So to diagnose this, you need the vesical fluid or scraping, skin scraping. Then you can do virus isolation, electron microscope, immunofluorescent test, IFT. Sometimes we acronym is IFT, sometimes with IF, it is the same thing. And then also you do the PCR analysis, and the control that vaccines are available, but not for many, let's say, herpes virus 1, you can have vaccine. Let's say equine herpes virus 1. They produce vaccine, but the vaccine is not effective. Because why? Even though animal have high antibody against the virus, they're still getting what we call this, the disease after sometimes. Okay. Hello? Hello, everyone.
Yes, bro.
Yes, bro.
Okay. I'm afraid that you are sleeping already. Okay, this is actually the diagram basically to share with you. Please remember this. Okay? Please focus on BHV1. I cannot give you so much BHV1. How? Because you study BHV1, you will see that there are lots of similarity of the disease, in equine, even though it is not the same virus. In equine, they have equine herpes virus 1. In bovine, they have bovine herpes virus 1. In, let's say, porcine, they have a porcine herpes virus 1. In cat, they also have feline herpes virus 1. All of them give a similar system. Okay, these viruses, no, feline cannot go to any other species, just what we call this in feline. Basically, these are actually the characteristics of the disease caused by the virus. These are talking about pathogenesis here, replication of the virus in epithelial cells around the upper respiratory tract. Then, in fact, in transmissions or infections of susceptible animal replication in epithelial cells. So you can see this one. It is a systemic cell-associated spread, meaning that the virus is transported in the system, many let's say organs, no, in the system, and then they are actually cell-associated. Meaning that they are inside the cell, and the cell actually are going everywhere, carry the virus. So that's why I call it cell-associated, not cell-free. It is not virus particle independently from cell going everywhere in the body and infect organ and cause problem to those organs. So it can cause encephalitis, encephalitis. So same thing in what we call this in horses. Equine herpes virus 1 also cause encephalitis. Abortion, yes, abortion here. EHB1, cause abortion. Same with BHB1. Okay. Cell associated, yes, EHB1 is the same too. So the difference is actually more towards species. Because now, horses, we're talking about running. Cattle, we're talking about produce meat and milk, not for running. So in horses, if they have this, let's say, infection, even the slightest, let's say, infection at the, what we call this, low level, horses will produce respiratory distress when they exercise and they run competition and so on. But cattle, you don't use it for competition like that. That's why you don't see that, regularly. You have to wait longer in order for you to see the relevant clinical signs enlisted here. So basically, it causes abortion, encephalitis, fatal disease in newborn, and also cause shipping fever. Shipping fever here means when you transport animals, animal got fever, and then you see what kind of, what we call these infections occur there. Okay, understand. So I hope that you all focus on bovine herpes virus here. Bovine herpes virus or IBR, infectious bovine rhinotracheitis, they are the same thing. Study that. Other viruses, which is I don't want to emphasize so much here, you can read on your own. You can also see this one, alpha-lafine herpes virus. This one actually in in a different species, no? It's not really, we don't really study here, this. So another one is ovine herpes virus 2 in sheep. So alpha-lafine herpes virus actually in wildebeest, we don't have it here in Malaysia. And then it is very similar with the malignant catarrhal fever. Malignant catarrhal fever, catarrhal means that what? It's inflammation, or inflame, redness and so on. So this actually only commonly occur in deer in Malaysia. So this is actually the thing. You can read on your own here, MCF. You don't have much time here. Okay. And then this one is actually the feline herpes virus in cat. It causes upper respiratory disease in cat, conjunctivitis, and very severe in cats. Okay. And then also you can see the persistent infection in cats, and then latency in cat, and then because of stress, the virus reactivated and produced, and what we call this transmit the disease, to other animals. So what about vaccines, for this feline herpes virus available? Okay. So you can see your own here, feline herpes virus, right? So you study this one, because now when you go to clinical year, you're going to see cats with eye problem like this. So you can suspect that, it is because of the herpes virus. You see here, in the earlier lecture, when I mentioned about diagnostics virology, what kind of test that you need to use? Immunofluorescence test. Okay? Virus isolation and identification using the swab and paired serum sample to detect the neutralizing antibody. So this is actually the usefulness of those, what we call this knowledge, basic knowledge about the how to do diagnosis. And you apply this kind of knowledge later on, in the clinical cases. Okay, you have equine herpes virus. I'm not going to come up with a question on equine this year, but basically you can see here how, let's say, virus pathogenesis of EHV. So EHV infect the endothelial cells in the lungs. Okay, from there it goes to cell-associated viremia. Then after that it goes to the, what we call this placenta, and and then transmitted to the babies, and then it can cause abortion. So that's how actually the virus causes abortion. So the virus normally in the lungs, respiratory tract, similar to BHV, whatever. So replicates in the, what we call this lungs, and infects the endothelial cells of the lungs and also the leukocyte, lymphocyte, and then the virus stay inside the cell, and the virus together with the cell transported to the endometrial vessel, the maternal fetal connections, and then in the end it causes abortion because the virus replicates in there. So this is actually the pathogenesis of abortion, just explained to you. Okay, the next one is Marek's disease virus. What is Marek disease? Okay, it is related. It is actually herpes virus of what we call this. In this case, is very special. It is not gamma. It is actually subfamily of alpha herpes virus, and it is related with the, normally gamma related with the, what we call this formations of the tumor. But Marek disease virus, of this one is special. It is alpha herpes virus, but Marek disease, and then it causes a problem in chickens. So be careful. This one could come out with what we call this in the exams later on, in the final exam. So Marek, other than BHV, Marek actually one another target here. It is actually slow CPE, produce CPE very slow, it takes longer time. It is highly cell associated, meaning that the virus like to stay inside the cell. And then the cell-free virus only you can see from the feather follicles. So meaning that the virus stay in the feather follicle. That's why if you suspect birds or chicken already infected by Marek disease virus, the sample that normally we take is actually feather follicle. We pluck the the feather at the base there of the feather. There is a follicle cell there. Then you take the cell, then we can send it to the lab for the test. There are three serotypes: mild to severe sign of lesion, apathogenic, and then related with the HVT. Herpes virus is actually T lymphocytes isolates. So it is a lymphoproliferative neurological disease of young chicks. Can cause unilateral paralysis and can cause ataxia. Ataxia is you can see kekejangan. I mean, the animal becomes half crazy because of this, because of the virus actually inside this in the nerve, and then the virus also infect the eyes, and then stable in follicle, where the follicle then there. So, meaning that the virus is transmittable through the, let's say, feather follicle. Follicle, of course, always drop around, so that could be source of infection or transmissions of the virus to other birds or chickens. So this is actually the pathogenesis here, virus from feather follicle, discrimination cells there, discriminations of cell, and then inhalations of this material, and then the virus replicates in the what we call this in the lungs and viremia, and go to lymphoid organs, busa, bone marrow, immunosuppressions, and they can cause transmissions of T cell and proliferation of T cell, and then also accumulations of inflammatory cell in that particular what we call sites, and then so as a result of that, what you're going to see is Marat disease virus. This one can cause neurolymphoma, meaning it can cause what we call this neuro is a nerve. So lymphoma, meaning that lymphocyte will be accumulated around the nerve. So around the nerve because of the virus actually there. So lymphocyte accumulated around nerve, and then accumulation in the nerve also, it causes enlargements of the nerve. So the nerve enlarged not because of the hyperplasia, it is because of the excessive accumulations of the lymphocytes there in the nerve. But you can see the size of the nerve becomes really big. So what kind of nerve actually? Is actually we're talking about femoral nerve, sciatic nerve actually related with this one. And then ocular lymphomatosis, it also can cause enlargement in liver, gonad, lungs, and skin, And then visceral organ may contain highly nodular. Basically, Marat disease virus cause enlargements of many, what we call this organ, visceral organ, because of the accumulation of the lymphocytes. Normally, it affects birds of 2 to 5 months, and normally not exceeding 32 weeks old, and high mortality is high, 80%. And the diagnosis, is actually based on the history of the bird's age, the bird age there. Not too young, not too old, not to get this Marat disease. If it is, you wouldn't see what we call this Marat disease virus in broiler chicken of 35 days. Because it's too young for the virus to replicate and show the problem. Because this virus need time to replicate and to come up with the disease, especially the tumor-like structure. So the clinical sign is very important for diagnosis. And findings due to enlargements of the visceral organ, could be kidney, could be liver, could be lungs. You can see become really big. And then the next one is to confirm Marat disease virus infection. You need to do immunofluorescence test, serology to see virus neutralization test, and the virus isolation is culture, and then use of CEM and USAC of 4DO for testing. So this actually to show enlargements of the visceral organ, including intestine. So this is another important virus here, avian infectious laryngotracheitis virus, ILT. So this one, you need to remember, ILT infectious the significance of this one is related with the chicken pox virus. Not chicken pox, sorry. Chicken pox is human. Fowl pox virus is a pox virus in chicken, fowl pox. So the characteristics of smallpox virus lesion is very similar to avian infectious laryngotracheitis virus infection. So they causes problem in respiratory system, and then common ulcer, and this actually virus can lesion they produce actually in the trachea is sort of the narrow the lesion, cause the narrowing down of the airways in the trachea, and it resulted, the chicken will have a difficulty in breathing. So why? Because of the multiplication virus on the epithelial cell lining the trachea. Same thing happened with the what we call this smallpox. So that's why I see, because of disturbance on the airline, the airways there. So you get the gasping, difficulty in breathing in the chicken. So when you see that signs, you may have a wrong misdiagnosis with the other virus. So these two viruses cause the occlusion or blocking of the airways. You can see this in the photo. The chicken actually open their mouth because of the difficulty in breathing. So basically, the virus through droplet infect chicken through inhalation, cause viremia, and then after laryngotracheitis, the tissue there become necrosis. There's some, affected tissue, can become necrotic, and necrotic material will block the airways. And because of sometimes, because too many, it form as good as another layer of membrane along the trachea. So that's how the trachea becomes, the airway so becomes narrowing down. So that what we call is another layer. We call it membrane. We call it diphtheretic membrane. It says, what is if I ask you a question, what is the diphtheretic membrane? Diphtheretic membrane is actually a tissue dye actually occur as a result of virus replication, and this actually tissue actually lining the trachea, lining the airways, and then as a result of lining there, it causes blockage or reduce, let's say, airways or airflow in the trachea. So the animal, the chicken open their mouth like that to get more air actually to the lungs. So this actually ILT, I think you need to read on your own here. So this actually diagram show you what is the diphthetic membrane. So this actually the common trachea. So then after that, the necrotic tissue accumulate here. So thicker and thicker, So in the end, the whole area becomes necrotic tissue, and only small airways actually in the middle left out. So this diphtheretic is a mixture of dead tissue, dead cell, plus with the mucus, and it becomes hardened after some time. Then that's it. So there is a vaccine available for the control of this disease, ILT, in chickens. Okay, this is a canine herpesvirus. I don't want to cover this one. I think it's too. You can read on your own. Very similar to herpes virus, Okay, pseudo rabies. This one is actually quite important. So the three viruses that I need you to emphasize. Bovine herpes virus, Marek disease virus, and pseudo rabies. It's actually a herpes virus in pigs. Okay, and then it causes inflammation in pigs. But this virus can be transmitted to other hosts and can cause pruritus. Pruritus means that itchiness. So animal become itchy, try to rub their body against a wall, against a tree, and so on. Because of the pruritus, inflammation of the skin. So it's caused by, let's say, pseudorabies. Another name, generic name of pseudorabies, actually or Jeski disease. It's same one. Pseudorabies or Jeski disease or porcine herpes virus 1. Same. They are same virus. They can cause reproductive problem. Cause mummified fetus, rebirth, weak piglet. So basically, the pathogenesis is very similar with the other alpha or herpes virus type 1. Virus replicates through, or enter the host through intranasal or oral, replicates in the oropharynx, travel into the nerve ganglia and cause inflammation of tonsil and so on. So, and then animal recover only with shed virus through saliva nasal secretion. And pseudorabies diagnosis based on sample from saliva. And yes, you have class or not after this? Hello?
Yes. Yes, Prof.
So
Yeah.
you have, I think, Prof had give you class, right? Give me a few minutes there. Then the control, you need to read this one. I'm not sure whether I need to have another time here. Maybe Monday, I think I will ask Dr. Yasmin for half an hour, to complete the other lecture here. So rabies is, you read this one. Rabies is it's quite interesting. What we call this thing to study. There are vaccine for the vaccinations of animals. They are gene deleted, and they use this vaccine actually to differentiate between, in future, to differentiate in the farm, vaccinated and non-vaccinated animal. There is a principle behind this one. So they use let's say vaccine that produce certain type of antibody. So without the antibody against certain viral protein, then you can say that, oh, this actually vaccinated, this animal infected and vaccinated, or this animal actually not infected but vaccinated. So by doing this one, you can differentiate infected animal, even though they already recovered, from the vaccinated animal but not infected. The reason we do this actually, if we like to come up with the disease eradication program, it is possible. Because now, you have to do that, you have to differentiate between vaccinated infected, because animal already infected, even they already infected, even though they recover, they still carry the latent virus. So you need to eliminate this animal. Okay, can cause abortion and so on, white horse range, and then and you have beta herpes virus, not so important, this one, and then not that important. So, and this actually a case study, a case virus now. ASEAN in ASEAN, elephant herpes virus in Asia, it only causes what we call this very mild disease, cold sore. But if this virus transmitted to African elephant, it can cause fatal hemorrhagic disease. Same thing here. If let's say African elephant herpes virus, let's say in natural host, the original host, it causes cold sore, mild problem. But when the virus transmitted to Asians elephant, it can cause fatal hemorrhagic disease. So this actually for you to know about the trans-species. When the virus from the natural host go to the species, it can cause big problem. Okay, I don't want to take your time here, but don't have much time here. For this one, just remember BHB, Maret disease, rabies. Remember that these three viruses under herpes. Pox viruses I haven't covered yet. Not much time left. So most likely I will take maybe 30 minutes of the time on Monday. Okay, first 30 minutes. So on Monday, I'm going to talk to Dr. Yasmin. Maybe we have to start early at 2 o'clock. And then 2:30, what we call this, we can stop. Or maybe if you like, we can have another, let's say, half an hour session tomorrow or another day. It's up to you. I mean, I need you all to let me know whenever you're free. Okay?
Sure, Prof.
Okay, you
Okay,
let me know.
Prof.
If you can give me, let's say, half an hour tomorrow, then it's good enough. So okay, thanks guys. You need to go for lecture another one, 11:59. I don't want to take other people's time. Okay, right.